Inside a temperature-controlled vault, historian Lindsay Chervinsky is showing me a letter written by a then 25-year-old commander of the Virginia Regiment, George Washington.
In March 2024, forensic scientist Courtney Cavagnino stared at her computer screen inside a military DNA laboratory at Dover Air Force Base. The likelihood ratio had just crossed 1 billion to 1. She ran the analysis again. Same impossible result. After 225 years of genetic silence, George Washington's bloodline had finally spoken, and it was about to rewrite everything historians thought they knew about the Founding Fathers' deepest ancestry. The secret in the data. Before we get to how scientists pulled off the impossible, you need to understand what that DNA actually revealed, because the findings didn't just confirm what historians expected, they challenged it. George Washington's paternal ancestry traces back to a specific genetic signature called haplogroup R-BY32422. That designation might sound like random letters and numbers, but it's actually an address on humanity's genetic family tree, an address that tells you where your direct male line ancestors lived thousands of years ago. And here's the catch. When researchers mapped where this genetic lineage appears most frequently in modern populations, the results pointed straight to the alpine region of Europe. We're talking Switzerland, northern Italy, southern France. In the Swiss canton of Ticino alone, over 53% of all men carry this same deep paternal signature. Not England, where Washington's family supposedly originated. The Alps. Population geneticists had studied RU152, the broader haplogroup containing Washington's specific lineage, for years. A 2020 Swiss population study found it reaching 26% average frequency across Switzerland. Researchers had traced it to Bronze Age Central European cultures, the Bell Beaker people, the Hallstatt culture, ancient Celtic and Italic populations who migrated across the continent millennia before anyone called themselves English. But that's not even the biggest surprise the DNA delivered. The genetic analysis also revealed something about the Washington family itself that made researchers double-check their data 3 times before believing it. The genetic relationships between family members came back closer than they should have been, approximately 1 degree closer than the carefully documented family tree predicted. The Washington family bloodline, it turned out, was far more tangled than anyone had ever realized. Generations of cousin marriages had folded the family tree back on itself, concentrating shared DNA in ways that historical records couldn't capture. So, how did scientists even get Washington's DNA in the first place? The man died childless in 1799. His direct genetic line should have ended over 2 centuries ago. That's where this story takes a turn nobody expected. The dead end. That wasn't. Here's the deal. George Washington never fathered a single biological child. He married Martha Dandridge Custis in 1759. When both were 27 years old, she was a widow who had already given birth to 4 children with her first husband, Daniel Parke Custis. 2 survived infancy. Proven fertility beyond any doubt. Yet, in 40 years of marriage to George, 40 years, Martha never became pregnant again. Not once. Not even a miscarriage in any historical record. Washington himself seemed genuinely baffled by the situation. In his letters, he attributed the childlessness to Martha. That's what men did back then. But modern medical researchers have a different theory. A 2004 study published in Fertility and Sterility suggested Washington likely suffered from tuberculosis-related infertility that blocked the pathways for sperm to exit his body. He'd contracted tuberculosis before marriage, and the infection may have caused permanent damage. Whatever the underlying cause, the result seemed absolutely final. When Washington drew his last breath on December 14, 1799, his Y-DNA, the genetic marker that traces direct paternal ancestry from father to son, should have vanished from the human gene pool forever. Basically, scientists hit an insurmountable wall. No descendants meant no DNA to test. Case closed. Except, it wasn't. Because here's what researchers finally realized, and it changed everything. Washington didn't need to have children for his Y-DNA to survive. He had brothers. NY DNA passes from father to son, completely unchanged. Think about it. George Washington and his younger brother Samuel both inherited the identical Y DNA from their father, Augustine Washington. If Samuel had sons, and those sons had sons, then Washington's genetic signature would keep flowing through history, generation after generation, completely independent of whether George himself ever reproduced. Samuel Washington absolutely did have sons. Those sons had sons, and their bones had been sitting in unmarked graves for nearly 200 years. The cemetery that kept its secrets. May 1999. Charlestown, West Virginia. A team of archaeologists, forensic specialists, and researchers gathered at Harewood Cemetery on the old Washington family plantation. The Harewood estate had been built by Samuel Washington beginning in 1770. George Washington visited his brother there multiple times during his lifetime. After Samuel's death from tuberculosis in 1781, the property passed to his son, George Steptoe Washington, who married a woman named Lucy Payne and raised a family. Around 20, Washington family members were eventually buried in the estate cemetery.
Just couldn't let go. It was really hard to let go. He had created this country. This country is his legacy, and he's worried about what's going to happen to it.
But there was a serious problem. Many of the graves had no headstones. The team arrived with ground-penetrating radar, excavation permits, and one ambitious mission: locate Samuel Washington's unmarked grave and recover DNA that might unlock the genetic secrets of the first president's ancestry. What they found instead was crushing frustration. The radar identified 5 burial sites showing soil disturbances. The archaeologists dug carefully through the clay, brushing away debris with painstaking precision. But what emerged from the West Virginia earth was fragmentary at best. Partial bones, incomplete skeletal remains. Nearly 2 centuries of bacterial contamination had ravaged whatever genetic material once existed inside those bones. And get this, the DNA extraction technology available in 1999 simply couldn't handle samples this damaged. Scientists tried. They processed the fragments, they ran analyses. The results came back useless, contaminated beyond interpretation. Unreadable noise instead of genetic signal. The team documented everything meticulously. The bones went into laboratory storage containers, and then they went home. For 25 years, the remains of Washington's relatives sat in climate-controlled storage, their genetic secrets locked away by the limitations of turn-of-the-millennium technology. But here's the twist that nobody saw coming back in 1999. The technology caught up, and when it did, everything changed. The military lab that cracked the impossible AFMES-AFDIL. That stands for the Armed Forces Medical Examiner, Systems Armed Forces DNA Identification Laboratory, located at Dover Air Force Base in Delaware. It's the Department of Defense's only dedicated human remains DNA laboratory. Their everyday job involves identifying fallen American service members, sometimes from conflicts stretching back to World War II, Korea, Vietnam. They work routinely with fragments that would defeat any civilian lab. They've built their entire expertise around degradation, contamination, and the supposedly impossible cases. When the Harewood Cemetery samples came to their attention, the team saw something beyond just old bones requiring identification. They saw an opportunity to validate their newest cutting-edge extraction techniques on historical remains that had completely defeated earlier technology. Lead researcher Courtney Cavagnino designed what they called a multi-marker approach, 3 separate DNA analysis methods running simultaneously, cross-checking each other. First, Y chromosomal DNA analysis to trace direct paternal lineages. Second, mitochondrial DNA sequencing to trace maternal connections. Third, a custom-designed hybridization capture panel targeting approximately 95,000 nuclear genetic markers that could reveal family relationships across multiple generations with mathematical precision. But all this sophisticated firepower meant nothing without a reference sample. Degraded DNA from 200-year-old bones is just chemical noise unless you have living DNA to compare it against. So Here's the deal that made the study possible. The research team reached out to Samuel Walter Washington, the current owner of Harewood Estate and a direct living descendant of the Washington family through the male line. Same name as his ancestors, same property in the family for over 250 years, same Y chromosomal DNA flowing through his veins. He agreed to participate. He submitted samples for comprehensive testing, including Big Y700 analysis through family tree DNA that examines over 500,000 markers along the Y chromosome, the highest resolution paternal lineage determination currently available. The comparison between ancient degraded remains and pristine modern DNA was about to blow the case wide open. The 1,000,000,000 moment. When the final results populated on Courtney Cavagnino's screen, the magnitude of what they had achieved hit the entire research team. 3 of the excavated burials matched to the living reference. Likelihood ratios exceeding 1,000,000,000 to 1. That's not a typo. 1,000,000,000 to 1 odds against these identifications being wrong. Burial 4, Lucy Payne Washington, the mother, identified through mitochondrial DNA and autosomal markers. Burials 3 and B, her 2 sons, Dr. Samuel Walter Washington and George Steptoe Washington Jr., confirmed through Y-DNA matching the living descendants' paternal lineage. And here's where the science becomes elegant. Those 2 brothers should have been impossible to distinguish genetically. They shared identical Y-DNA from their father and identical mitochondrial DNA from their mother. Same fingerprints on both major lineage markers. But the team had a third tool, autosomal DNA, the nuclear genetic material inherited from both parents that shuffles differently with each generation. By comparing how much autosomal DNA each skeleton shared with the living Samuel Walter Washington, they could calculate precise degrees of relatedness. The living Washington proved to be a 4th degree relative to one brother and a 5th degree relative to the other. Different amounts of shared DNA. Different identities confirmed with complete scientific certainty. For the Washington family, the remains could finally be properly memorialized after 200 years of anonymity. But for historical understanding, the implications were only beginning. If you're finding this investigation fascinating, hit that subscribe button right now, because what the DNA revealed about Washington's deep alpine ancestry and the mathematically proven tangled family bloodline is even more surprising than the identification itself. The alpine signature. The living Samuel Walter. Washington's Big Y700 test came back with a precise haplogroup classification. RBY32422. Now, you need to understand what that technical designation actually means, because this is where historians started raising their eyebrows. Y-DNA haplogroups function like addresses on a massive family tree, encompassing all humanity stretching back hundreds of thousands of years. By identifying someone's specific haplogroup, scientists can trace where their direct paternal ancestors, father to father to father, going back millennia, originated in the ancient past. R-BY32422 falls within a broader genetic lineage called R-U152. And when population geneticists map where R-U152 appears most frequently in modern populations, you don't find the highest concentrations in England. You find them in the Alpine corridor. A comprehensive 2020 Swiss population genetic study confirmed RU152 as the single most common Y-DNA haplogroup among Swiss men, averaging 26% frequency nationally. In the Italian-speaking canton of Ticino, nestled south of the Alpine peaks, over 53% of all men tested carry this same paternal signature. Northern Italy shows similarly elevated frequencies. Southern France, parts of Germany. Ancient DNA studies have linked RU152 lineages directly to Bronze Age Central European cultures. The Bell Beaker expansion, the Hallstatt culture, Iron Age Celtic and Italic peoples who migrated across the continent thousands of years before the English language existed. So, here's the catch. Washington's documented family history traces to England. Generations of Washingtons lived there before crossing the Atlantic, but his deep paternal DNA signature points toward continental Europe, the alpine region where prehistoric populations lived millennia before historical records began. The DNA doesn't contradict English ancestry. RU-152 appears at meaningful frequencies in southern England, probably carried there by ancient migrations. The genetics and genealogy remain compatible, but the DNA adds a dimension no document ever captured. Washington's male line ancestors, going back thousands of years into prehistory, originated somewhere in the heart of Europe, not the British Isles as a starting point, the Alps. First president, first genetic confirmation, a story older than anyone imagined. The family tree that folded. But here's the revelation that genuinely made the research team pause and verify their calculations multiple times. When Cavagnino and her colleagues calculated the exact genetic distance between the living Washington descendant and the historical remains, something in the numbers looked wrong. According to meticulously documented genealogical records tracing every generation, the relationships should have landed at specific predicted distances. They didn't match. The DNA consistently showed family members were approximately 1 degree closer genetically than the paper family tree indicated. The researchers ran calculations again. Same result. The explanation clicked. Pedigree collapse. And get this, it wasn't some shameful scandal being exposed. It was simply how elite colonial American families routinely operated. Cousin marriages were strategic tools in early America's upper classes. They preserved family property from fragmentation. They maintained social standing. They concentrated wealth. When first cousins marry, or second cousins marry repeatedly across generations, descendants inherit overlapping DNA segments from multiple shared ancestors rather than independent genetic contributions. The mathematical result, DNA that's more similar than a standard family tree would predict. The Washington family tree hadn't just branched across generations, it had folded back on itself repeatedly, accumulating shared genetic material through consanguineous marriages that historical records documented but never quantified. Historians always knew these families intermarried extensively. Now, there was hard mathematical genetic evidence revealing exactly how tangled those colonial bloodlines became. The Washingtons weren't just related by marriage, they were genetically denser than their documented tree could indicate. The DNA told a truth the documents only hinted at. The First Lady's sister. Here's a connection demonstrating how deeply intertwined America's founding families truly were. Lucy Payne Washington, the mother positively identified in this study, had a younger sister named Dolly Payne Madison. Yes, that Dolly Madison, the future First Lady of the United States, the wife of President James Madison, the woman who famously insisted on saving George Washington's portrait when British troops burned the White House in 1814. And get this, James and Dolly Madison were married at Harewood itself, in Lucy's parlor, on September 15, 1794, the same property where Lucy lived with her Washington husband, the same grounds containing the cemetery where her bones would rest for over 2 centuries before DNA. Science could speak her name. Lucy later remarried after her first husband's death. Her second wedding, to Supreme Court Justice Thomas Todd, took place March 29, 1812. The venue, the White House. It was the first documented wedding ceremony ever held in the president's mansion. Her sister Dolly, then First Lady, hosted the Washingtons, the Madisons, the Supreme Court, the First Ladies, all bound through marriage, property, blood, and now irrefutably confirmed through DNA. The technology that will save thousands. This study wasn't purely an exercise in satisfying historical curiosity about a founding father. The techniques validated on the Harewood remains have immediate, profound, life-changing applications for thousands of American families who are still waiting for answers about their own lost loved ones. AFMES-AFDIL identifies fallen service members as their primary mission. That's what they do every single day. Their active caseload includes remains from World War II, Korea, Vietnam, and every conflict since. fragmentary bones and teeth that have been waiting for positive identification for decades. Here's the critical point. Some of those military samples are actually in worse physical condition than these 200-year-old Washington family remains proved to be. Senior study author Charla Marshall, deputy director of the laboratory, stated it plainly to media outlets. The multi-marker approach validated on these historical fragmentary bones can now be confidently applied to military identification cases that previously seemed completely hopeless. The methodology works. The science is proven. Think about what that means in human terms. Families who have been waiting for closure since the 1940s. Sons, fathers, brothers, husbands, whose names were never scientifically confirmed. Graves marked unknown in military cemeteries from Hawaii to Europe, from the Pacific to the Korean Peninsula. The Harewood study dramatically expanded what's technically possible for bringing those service members home, at least in name. For the genetic genealogy community researching their own ancestry, the implications are equally massive and immediate. The Washington Surname Project, maintained at Family Tree DNA, contains nearly 200 participating members, representing over 40 different predicted Y haplogroups. Now, anyone claiming paternal descent from George Washington's family line can submit their own carefully tested DNA and compare it directly against the confirmed presidential haplogroup. Some researchers will find their cherished family legends scientifically validated. Their Washington ancestry is real, proven by genetic evidence that can't be fabricated or misremembered. Others will discover uncomfortable truths, that family stories passed down for generations don't actually survive genetic scrutiny. Either outcome represents truth. Either way, the genetic record is now definitively open for anyone willing to test. The father who left no children. George Washington once wrote to a nephew that he possessed a moral certainty of dying without biological issue. He believed it completely. He had resigned himself to it. He was absolutely right about his direct line ending, and he was completely wrong about his genetic legacy disappearing. His direct descent ended on December 14th, 1799, the day he died at Mount Vernon. No biological children of his own, no grandchildren, no great-grandchildren carrying forward his direct bloodline through father-to-son transmission. The man who fathered a nation never fathered a child. But his Y-DNA never died with him. Not even close. It flowed silently through his brother Samuel's lineage, generation after generation, century after century, decade after decade, carried by grandnephews who lived and died with absolutely no awareness of the historical significance coded into their chromosomes. Preserved in fragmentary bones beneath West Virginia clay in unmarked, forgotten graves. Waiting with infinite patience for the precise moment when technology would evolve far enough to trace that genetic signal definitively back to its presidential source. Today, Samuel Walter Washington owns Harewood Estate, the identical property where his ancestors built a home beginning in 1770, where George Washington visited his brother, where James Madison married Dolly Payne, where the dead kept molecular secrets until science learned to listen. The remains of Lucy Payne Washington and her two sons can now receive proper memorialization after 2 centuries. The genetic identity of America's first president has been scientifically confirmed for the first time in history, and the tangled family tree of America's founding generation looks considerably more complicated than any textbook acknowledged. Washington was the father of his country, the father of the Continental Army, the father of constitutional government, and now, 225 years after his death, we know with scientific certainty he was a genetic father too. Not through children of his own blood, but through shared Y chromosomal inheritance from his brothers. The quiet persistence of DNA across 10 generations. And forensic scientists who refuse to accept that some mysteries stay buried forever. The father of the nation left genetic descendants after all, just not in the way anyone ever expected. If this investigation blew your mind, smash that like button and subscribe for more deep dives into hidden DNA secrets of history's most famous figures. What other founding fathers might be concealing genetic surprises beneath their marble monuments? Drop your theories in the comments. I read every single one.